have Amy Zwierzchowski-Zarate from Dr. Mark Diamond's lab in the Center of Alzheimer's and Neurodegenerative Diseases. Amy is in the neuroscience program. She leapt from the corporate ladder to a calling in science because she wanted to solve problems that deeply impact humankind. Her talk is entitled When Good Tau Turns Bad, Uncovering the Origin Story of Alzheimer's Disease.
Do you ever wonder what turns a good thing bad? Like, how does Darth Vader happen? Supervillains have the best origin stories. We have this need to understand and rationalize what drives a young Anakin Skywalker to transition to the dark side. But it's also useful for understanding neurodegenerative disease. The squiggly line under Anakin is a tau protein, and the tau protein can exist in two states, good and bad. In its good state, it's wonderfully existing inside of our brains. However, it can encounter something that causes it to shapeshift, turn bad, and wreak widespread havoc and atrophy on the brain, like that seen under Darth, and also seen in the brains of patients with Alzheimer's disease. If we could pinpoint the origin story of tau's transition to Alzheimer's disease, maybe we could find ways to revert it back to good. And that's what my project aims to do. Alzheimer's disease is largely sporadic in origin, meaning we don't know what causes it. But we do know that tau is involved, and we know that when tau shapeshifts is when people start to show symptoms. Tau is positively charged, and when it encounters negatively charged molecules, it shapeshifts as well. A lot of the things we use to do this in the lab, however, are unlikely to be doing this in the brain, except for one, RNA. Using biochemistry, as well as a cellular technique that we've developed in our lab, I can ask questions about what changes tau from good to bad. Our cellular technique takes fluorescent proteins, tags them to tau, and when tau is good, the cells light up and it's diffused everywhere. When tau turns bad, you start to see clumping and aggregates form throughout the cells. To systematically understand how RNA could be converting tau, I took different sequences of RNA, mixed them with tau, and asked whether they turned to tau bad. While all of the sequences bound tau with similar affinity, only some of the sequences turned tau to bad. I wondered if removing the RNA could turn tau back to good. By digesting the RNA away with nucleases, I saw tau revert from bad back to good. I wondered if this could be happening in brains. When I take tau from an Alzheimer's brain and add it to my cells, I see widespread clumping and aggregation throughout all of the cells. But if I treat that tau with nucleases, I see 90% of those cells revert back to their happy, diffuse, good state, suggesting that RNA is involved in the origin story of tau's transition to Alzheimer's disease. And this is what I'm investigating. But I'm not stopping with Alzheimer's disease. I forgot to mention, but tau is involved in 20 other diseases called tauopathies. And if I can find the origin story of tau's transition to cortical basal degeneration or frontotemporal dementia, maybe I can find ways to change it back to good, or prevent it from ever transitioning to the dark side in the first place.